Previously, we created a script which only preprocessed the data. In the current tutorial, we will combine both preprocessing and model estimation into a single script using ubersubject.py. The first thing you need to do is create timing files, also known as onset times, which indicate when each trial occurred. Use a terminal to navigate to the directory containing all of your subjects. Then go to this GitHub page and either download the script or copy and paste the code into your terminal. Once you've done so, press Enter to run the code. After a few moments, timing files for both conditions will be created for each subject and placed in their corresponding func directory. Take a look by typing cat sub08 func incongruent.1d. There will be two rows, one for each run, with the onset of each trial indicated in seconds. Let's start with a clean slate by navigating to the sub08 directory and typing rm-r subject_results. Then type ubersubject.py and press Enter. First, fill in the fields like you did during pre-processing, selecting the anatomical data set and the functional images. Now, let's look at the fields relevant to creating the model, beginning with stimulus timing files. Click on the Browse Stim button and select the files congruent.1d and incongruent.1d, located in the sub08 func directory. When you click OK, you'll see a table populated with the timing files you selected. In addition to showing the name of the timing file that you selected, there are three other columns named Label, Basis, and Type. Label is how the timing file will be referenced in the command that does the model fitting, whereas basis and type specify which basis function to apply to the timing files. The default basis function of gam specifies that the onset times should be convolved with the canonical href, which is shaped like a gamma distribution. This basis function requires that only one parameter be estimated, namely the height of the href, which corresponds to the amount of neural activity in response to that condition. Symbolic GLTs specifies general linear tests for the beta weights estimated for the conditions above. Click on the Init with Examples button to show example syntax for setting up the tests. When you do, you will see two contrasts, I-C and mean.ci. Let's modify this table to set up the contrasts that we want. In the first row, change the label column to incongruent minus congruent. And in the symbolic GLT column, leave them as they are. Likewise, in the second row, specify a contrast label of congruent minus incongruent. And in the symbolic GLT field, type congruent, then a space, that's important, minus incongruent. No sign implies a contrast weight of +1, while a negative sign indicates a contrast weight of -1. Therefore, the line congruent, negative incongruent means to weight the parameter estimate for the congruent condition with +1, and to weight the parameter estimate for the incongruent condition with -1, and then to take the difference between the two. We will look at these contrast estimates once the model has been estimated. For the extra regress options, change the number of jobs to 8, or whatever number of processing cores you want to use. Uncheck the Run Cluster Simulation box, since we'll not be reviewing that here. Then check the Execute 3D RemelFit option, as this will create a separate statistical dataset that better accounts for temporal autocorrelation than the traditional 3D deconvolve approach. This will be discussed further in a video on group-level analysis. Also, change the extra align options and the extra TLRC options, just as we did in the pre-processing video. Now run the script by clicking on the same three buttons that we clicked to pre-process the data. Before pressing go, don't forget to make the same changes to the script as before, such as init xform after the auto-TLRC command. In this script, the data will first be processed, and then a model will be estimated. The total processing time will be about 5 minutes. When the script finishes, navigate into the folder sub08 subject results group.flanker sub08 sub08.results. In addition to the preprocessed blocks you saw previously, you will now see statistical data sets. The one labeled stats.sub08 has been analyzed using the traditional 3D deconvolve approach. The dataset stats sub08 REML has accounted for variability in temporal autocorrelation. You will also see a few files beginning with an X, such as x.xmat.1d. These represent different parts of the design matrix. For example, you can view the design matrix by typing AIV x.jpg. The first three columns are the drift regressors, which will capture linear and quadratic trends in signal. The two columns in the middle are the task regressors, with dark bars indicating when a trial occurred. And the last six regressors are the motion parameters. Notice how the drift and motion regressors are modeled separately for each run, with time running from top to bottom instead of the traditional left to right. We are now ready to look at the contrast maps of our data. Type Afni to open the GUI, and select ANAT-Final sub08 as the underlay image. For the overlay, select stats.sub08. You should see something like this. The most noticeable features of the now opened Define Overlay panel are the slider bar, which allows you to threshold the images to only see values above a certain number and masks out everything else. There's also the ULay, OLay, and THR menus corresponding to the underlay, overlay, and threshold subbricks. Let's begin looking at the slider bar some more. If you move it up and down, you will see voxels either disappear or reappear. That's because we are thresholding, or removing, voxels that fall below the threshold number to the left of the bar. This number is based on the subbrick that is selected in the THR menu. In this case, the subbrick that was selected for us when we opened the viewer was volume number 2, the t-statistic map of the beta weights for the incongruent condition. As you move the slider, the number below the slider bar, p equals, changes as well. This will only show voxels in this case that pass a p-value threshold of 0.0271. Now change the OLay subbrick to incongruent minus congruent coef and the THR subbrick to incongruent minus congruent tstat. Then set the uncorrected p-value threshold to 0.05 by right-clicking the p equals text and entering 0.05, and then pressing enter. Then click around the brain, observing where the statistics are positive and where they are negative. Where do you notice significant clusters of activated voxels? Are they where you would expect them to be based on the paper? Now that we've analyzed a single subject from start to finish, in the next video, we'll learn how to create a script to automate the same analysis for all of our subjects. Are you excited? I'm excited. After you've pre-processed and set up a model for a single subject, you'll need to do the same steps for all of the subjects in your data set. This may seem tedious but doable. We have only 26 subjects and only 2 runs per subject. An alternative, however, is to script your analysis. Just as an actor has a script which tells him what to say, where to stand, and where to move, so you can write a script that tells your computer how to analyze your data sets. This has the double benefit of automating your analyses and being able to analyze data sets of any size. The code for analyzing 2 subjects, or 200, is virtually identical. First, we will create a template that contains the code needed to analyze a single subject, and then we will use a for loop to automate that analysis for all of the subjects. When you analyzed sub08, you created a file called proc.sub08. This contained a list of Afni commands composed in a manner determined by the ubersubject.py GUI. From the flanker directory, copy this file to the current directory and rename it to proc.flanker.sh. We will then need to ensure that we can loop this script over all of our subjects without error. And to do that, we will need to edit the script. To begin, open the proc_flanker.sh file in a text editor such as Text Wrangler. Scroll to the conditional statement on lines 26 through 31. If the user provides an input, then the variable subj is set to whatever that argument is. In other words, each variable $subj in the script will be replaced by the input argument. However, there are other instances, usually involving paths, that still have the string sub08 hard-coded into them. In order to make the script more flexible, we'll need to replace these with the subj variable. If you are using Text Wrangler, first go to the menu at the top of the screen and then click on the Search menu. From here, select Find. Search for the string sub-08 and replace it with $, curly braces, subj. Next, we will need to replace any absolute paths with a relative path. As you can see in the script, there are several lines of code that contain paths, starting with users aeon desktop flanker. We will replace this with the $pwd variable, which is a shorthand for the path to the current working directory. This will ensure that the script will be adapted to the current computer's directory structure, and that no errors will be thrown due to the script being unable to locate where certain files are. Search for the string users aeon desktop flanker, or whatever the name of the path is, which points to the current directory containing your subjects, and replace it with $, curly braces, pwd. Also replace line 255, the path users aeon a global, or whatever your username is, with ~/abin, which will point to your Afni library. We will now use this updated preprocessing script in a for loop to analyze all the subjects in our data set. Use this for loop for I in backticks cat subject list, tcsh proc flanker, $i, and then move $i results to the $i directory. This will run the script proc_flanker.sh for each subject in the file subjectlist.txt, using each consecutive line in the file as an argument each time the script runs. Each analysis will take 5 to 10 minutes, depending on the speed of your computer. Once we've completed the analysis for all of the subjects, we can do a group-level analysis to determine whether our congruency effect generalizes to the population. Our goal in analyzing this data set is to generalize the results to the population that the sample was drawn from. In other words, if we see changes in brain activity in our sample, can we say that these changes will be likely seen in the population as well? To test this, we will run a group-level analysis, also known as a second-level analysis. In Afni, this means that we calculate the standard error and the mean for a contrast estimate, and then test whether the average estimate is statistically significant. We will do this group-level analysis in two ways, using 3D t-test, which uses only the contrast estimates, and 3D MIMA, which accounts for both the difference between the parameter estimates and the variability of that contrast. Just as we used a graphical user interface to preprocess the data, we can also use a GUI to set up our group-level analyses. If you type the command uber-ttest.py from the command line and press return, you will see this window. In the first field, Program allows you to choose between 3D t-test and 3D MIMA. In both the script name and dset prefix fields, enter flanker_inc-con_t-test. For the mask dset, you can choose any one of the subject's mask, group, plus TLRC images located in the results directory, as they should be similar across all the subjects. We will now select each of the subject's statistical data sets using wildcards. If you click on the button Get Subject Datasets in the Datasets A section, you will be prompted to select a representative stats dataset. Select any of the subject statistical datasets and replace the last two numbers of the subject ID with 2 question marks. Once you have finished doing that, you can then click on Apply Pattern. If all the subjects were analyzed the same way and have the same directory structure, you should see 26 entries in the field below. Review them to make sure they are all there, and then click OK. At the bottom of the Datasets A section, you will see a few additional fields. In Set Name, Group or Class, write Inc-Con, and in Data Index Label, type 7. Why 7? If you open a terminal and navigate to any of the subject's results directories, type the following: 3dinfo-verb, followed by the name of the stats dataset. This will show all of the subbricks in that dataset along with their labels. In our current example, we are looking for the contrast estimates for incongruent minus congruent. The output of 3dinfo shows that it's located in subbrick 7. As with the uber subject.py script, there are buttons at the top of the GUI for both generating the script and then running the script. First, click on the icon that looks like a sheet of paper, which will show you the command that has been generated. Review it to see how it has inserted all of your inputs into a command called 3dttest++, which will run the actual group-level analysis. Then click the green Go button. This only takes a second. When it has finished, go back to your terminal and type ls. You will see a new directory called group_results, and within that a folder called test00-3dttest++. Navigate into that folder, which you will see contains the script that was used to generate the results, flanker inc-con_t-test, and then another folder called test.results, which contains the group level output flanker_inc-con_t-test plus TLRC. Load that into the Afni viewer and overlay it on top of the MNI 152 average template. Threshold these images to an uncorrected p-value of 0.001 by clicking on the p = sign underneath the slider bar. This allows you to specify a specific p-value, uncorrected, for each voxel in the brain. Then clusterize these results to only show clusters with an extent of 40 voxels or more. This will create an image like the one you see now. As an aside, in the text box down below, there's a link to a chapter explaining how to perform cluster correction to determine whether these cluster sizes are significant. Does the location of the activation make sense to you, given the task and the paper this experiment was based on? We can run a similar procedure with 3D-MIMA, this time selecting both the beta weight and the corresponding t-statistic. We will change our statistics datasets to the ones ending in REML, or restricted maximum likelihood. Subbrick 7 in this case is the beta weight, and subbrick 8 is the t-statistic. You will run this analysis virtually the same way as you did the analysis for the t-test. You will see a window open up tracking the progress of each slice that's being analyzed. This should only take a few minutes. When it has finished, you will see a new directory in the group results folder called test002-3D-MIMA with a subdirectory called test results. Navigate to that folder and overlay the results as before. Are the effects in the same location? Do the effects look stronger or weaker, and why? Now that you have finished group level analyses, you are ready to perform more focused analyses using regions of interest. All that and more in the next video. A region of interest, or ROI analysis, is a method of extracting data from a subset of voxels, also called a mask. One way to create a mask is to use an atlas, or a map that partitions the brain into anatomically distinct regions. Another technique is called the spherical ROI approach, in which a sphere of a given diameter is centered at a triplet of specified x, y, and z coordinates. These coordinates are often based on the peak activation of another study that uses the same or a similar experimental design to what you are using. In this tutorial, you will learn how to do both types of region of interest analysis. Afni comes with several atlases in both TeleRac and MNI space, which can be accessed through the Afni GUI. Navigate to the sub-01 results directory and open Afni. Select ANAT Final sub-01 as an underlay, then click on Define Data Mode and click on Plugins. From the drop-down menu, select Draw Dataset. You will first need to click on the button Choose Dataset for copying. The purpose of making a copy of that dataset is to create a clean dataset with the same dimensions as the other images, but which we can write on by marking whichever voxels we want to belong to our ROI. Since all of our data has been normalized to the MNI average 152 template, we are able to use one of our normalized anatomical images. Select the atlas DD, Deci, MPM, and then click on the drop-down menu below it. You have many different regions to choose from, but for now, select cortex, LH, G and S, gyrus and sulcus, cingulate, mid-anterior, and then click on the button Load Infill. This will highlight in red all of the voxels belonging to that region of the atlas. You can undo this by clicking on the Undo button, which keeps several steps in memory. Now, right-click the area to the left of the Label drop-down menu to open a more compact view of the atlas regions, and select Cortex, RH for the right hemisphere, G and S, cingulate mid-anterior. As before, click on Load Infill to add that region to the current mask, and then click Save As. Call this mask Mid-ACC. When you are finished, click Done. Before we can extract data from this mask, we will need to match the resolutions of the mask dataset and our statistics dataset by using Afni's 3D resample command. This command requires both a master dataset, which we will be resampling to, and an input dataset, which will have its dimensions and resolution changed to match the master dataset. For the prefix option, type mid-ACC _ RS plus TLRC. This will create a new file, mid-ACC-RS, in which RS stands for resampled. Move this mask to the subject directory by typing mv mid-ACC-RS ... We can then use this mask to extract data for our ROI analysis. For the current experiment, we are interested in extracting the beta weights for the congruent and incongruent conditions separately, so that we can see what is driving the effect of the contrast incongruent minus congruent. To see which subrecs we will need to extract, from the flanker directory, type 3dinfo-verb sub-01 sub-01.results stats.sub-01. This will return a list of all the beta weights and contrast weights contained in the stats file. Note that subrec 1 corresponds to the beta weight for congruent, and subrec 4 corresponds to the beta weight for incongruent. The individual subrecs can be extracted using the following code available on my GitHub. You can either download this file, or you can simply copy and paste the code by clicking the Raw button, and then put it into your terminal and press Enter. This will put each subject's beta weights for each condition into a separate file of congruent and incongruent. You can now extract data from the anatomical mask by using the 3D mask av command. The quiet option suppresses irrelevant information. We also specify the mask and the betas we're extracting from. We do this for the incongruent betas as well. You can then copy and paste these beta weights into a program such as R. I like to use the scan command in order to copy and paste a list of numbers into a variable. In this case, I'm putting them into the variables con and inc. You can then run a paired t-test to see whether the result is significant. But what if we want to base our ROI on another paper's results? For example, let's say that we read a paper reporting cognitive control effects with peak activation at the MNI coordinates 0, 20, 44. We can create a 5 mm sphere centered at these coordinates by using the following code with Afni's 3D undump. The SRAD option specifies how large the radius of the sphere will be, while the master option creates a mask dataset with the same resolution and voxel size as the master dataset. The prefix option labels the output file, and -XYZ specifies the coordinates around which to center the sphere. The dash after the XYZ option indicates that the output on the left side of the pipe, in other words, echo 02044, should be used as input for that option. The result of this command will be a file called conflict ROI, which you can use for ROI analysis. For this, we will use the same 3D mask av command as we did in the previous ROI analysis. The output will be 26 rows, one number per row, representing the average beta estimate across the voxels of the mask that we extracted from. Use the same command to extract the beta estimates for the incongruent betas file, and then copy and paste both sets of numbers into a package such as R. Here I'm going to use the same steps as we used for the anatomical ROI analysis. The numbers you get from this analysis should look different from the ones you created using the anatomical mask, which reflects our more targeted analysis approach. Now that you've completed most of the analyses you would do with a typical fMRI dataset, we will move forward to a more advanced analysis known as surface-based analysis using SUMA, the surface mapper.